This is my lord, for the record, Ellison Karhanga. Ms. Hingley, just to remind you, you're still under oath. We had ended when you were explaining the DNA profiling process. And I just want you to help take this coach to your report and take us through that process. What you had started, but just confirm if there's any part of your report that explains this process.
The appendix of my report contains details of the DNA profiling process.
So if you could just take us to the appendix. Profile for DNA.
Sorry. So the techniques we use look at specific regions of the DNA which are known to vary widely between different individuals. DNA profiling techniques can therefore be used to conclusively exclude someone as the donor of an unknown source of DNA and may provide very compelling evidence of association, but they cannot prove proof of identity. The standard DNA profiling technique used in this case is a sensitive DNA analysis technique called STR DNA profiling, or short tandem repeats.
So it's a short tandem repeats
Yes.
means STR.
Yes. This technique of DNA amplification, in which specific regions of DNA are targeted and copied many times. This increases the amount of DNA available for analysis and means that an SDR profile may be obtained from samples where the initial quality of DNA is unsuitable for other DNA profiling methods.
So okay, maybe just take us through Verifiler Plus, and then you'll tell us, then I'll ask you a few questions just to make it clear for all of us.
So the technique used here in Uganda is Verifiler Plus. This technique,
Please speak into the microphone.
we're just switching over. So this technique is optimized for small amounts of DNA.
Okay.
It amplifies or copies 25 different areas of DNA. The individual components of an SDR profile, technically known as alleles, are represented as a series of peaks which can be measured and given a numerical designation. A person will have 2 alleles for each SDR, one inherited from each parent, unless the same SDR is inherited from both parents, where only one allele will be seen.
Okay, so in simple terms, if we go back to the DNA sample of Mole Katanga,
Yes.
you'll show us that, okay? And that, my lord, was in Defense Exhibit 5.
Sorry?
That document is in Defense Exhibit 5. The
EGC, electropherogram.
the electropherogram and page 13.
Yes.
Where did you get that document from?
So this document is the one I've referred to as the DFS confidential file transit ladder, which was supplied by KAA and referred to in my statement.
And on what page can we find Molly Katanga's DNA?
Molly Katanga's DNA profile starts on page 13.
Okay. Okay, so now you had explained to my lord about Verify Laplace looks at 25, is it 23?
There's 25 different areas,
Okay.
one of which is the sex determining test and one of which is specific to male DNA. So there's 23 SDRs sites, 25 in total over the profile.
So, can you show us what you mean by an STR site?
So on the graph, in the boxes above what you can visually see as a peak, these are the individual areas of DNA which are targeted.
So that's the box which starts with what number?
In this system, the first area is D3. And in this area, you can see that there's only visually one peak, one piece of information. But
Where you find one piece of information, is there a technical term for that?
so technically, when you see one piece of information, this is known as a homozygote. So this is where the child has inherited the same piece of information from both mum and dad.
So, and when you talk about, you told us about piece of information, and in your document you talked about allele.
Yes,
there a
allele
difference?
is the technical term. Allele is just the variation of information within the certain area of DNA.
Okay, so what is the difference between piece of information and allele?
They refer to exactly the same. Allele is just the scientific term, where piece of information is just a description used for non-scientists.
Now, so where's the next STR location?
So the next one on this system is known as VWA,
Okay.
where you can clearly see there are two pieces of information visually.
And then there's a number beneath them.
Yes, so
Can you
underneath
explain
each,
the numbers beneath each?
so underneath each piece of information, you have then two numbers. The first, which in this case 15, merely relates to the number of times a specific sequence of DNA is repeated. So in this instance, this piece of information has been repeated 15 times, and the next one is 17.
Okay, I'm not going to go through all of them, but if you can just explain. So, in the boxes up there, we have the pieces. We have what did you call those in the boxes? You call them STR something?
So the labels here are the different areas of the DNA molecule which are specifically targeted by the DNA profiling technique.
Okay.
These are the areas that are known to vary greatly between individuals.
Okay. Now, if you just go up a bit to the next, we're seeing one line which ends at D6.
D6, correct. The next one, which is green, begins on the left-hand side. You can just see this area here is a specific male-only DNA test.
And what is it called?
So this one is YINDEL. So obviously this is Molly's DNA profile. Molly being a female, there will be no information here because only a male can produce information.
Okay.
The next one is Amelo. This is the sex.
Just a moment. The first one, YINDL, has no information.
Yes.
And where do we see the lack of information?
How can you tell that there's no information?
Interpreting that.
So if we look, there's two red markers here, this one and this one. If it was a male DNA profile, I would expect to see a visual representation as the rest of the graph. So there will be a single peak here, which indicates a male.
Okay, let me ask it like this. You have in the first you have red markers throughout.
Yes, the red markers are purely demonstrating the boundaries of each of the areas of DNA.
So at the first location you have the red markers, the two red markers represent the two boundaries for the Y-indel. Would I be right to say that?
That's correct. So if there was DNA from a male present, there would be a piece of information within these red markers.
Okay, and then at the next
one, this is the sex determining case, Amela Jenin.
Okay.
And as we can see, we have one single peak here. This is because Molly is a female. Females inheriting the X chromosome only from both parents. Hence one peak here indicating female.
And the next location, which is D8, we have
D8, again, you are seeing two pieces of information, meaning that Molly has inherited two different pieces of information from mum and dad.
okay, and so we are seeing two pieces of information throughout the second line, right?
Correct.
And then if you go to the third line,
Exactly the same principle, two pieces of information at every area.
now we're also seeing different colors. I'm seeing red, I'm seeing green, I'm seeing
Yes,
black.
these are merely fluorescent tags added during the DNA profiling process in order for us to visualise the DNA profile. Without these colours, the graph would just be a mash of information and we couldn't separate it out visually as we do now.
Now I want to wind up on that brief introduction to DNA profiling by talking about the profile of Mole Katanga.
Yes,
If I was to write the DNA profile of Mole Katanga
yes.
without your graph and without your 6H2Y, if I was to write Mole Katanga DNA profile and just to write it across a piece of paper using this,
Yes.
what would it look like? How does that?
In effect, it is just a sequence of numbers with each number representing a specific area on the DNA molecule.
Okay, so basically, would I write the different locations?
The different locations, the DNA profiler is in a very specific order.
Okay, now, so I have my 23.
Mr. Palmanga.
Yes, much obliged.
Miss,
Hindi.
Mole Katanga's DNA profile will be a sequence of numbers with each number representing.
each individual piece of information, but those numbers are in a specific sequence and must be attributed to the area of the DNA molecule.
So, without, for example, commenting on the accuracy of what I'm going to tell you, well, just for purposes of understanding, if I go to the DNA report, yeah?
Mm-hmm.
Is there, on the DNA report, can I find Mole Katanga's DNA profile without these, just written like in a straight line?
Yes, there was a table of the pieces of information of all the DNA profiles obtained. However, that table didn't have every single area that Verifiler Plus uses. It appears that the information taken over relates to a system global filer. So the information on the electropherogram is not totally within that table on the DNA report.
But do you have a copy of the table?
I do.
So you're saying that the table just quotes fewer locations. Have I understood you correctly or have I misunderstood you? I just want to know if I've understood you.
That's correct. So the information, you will notice there is a difference between all the information on the electropherogram in comparison to the table. 2 to 3 areas haven't been carried over.
To the table. Okay?
Just one second.
That's the table.
Yes, this is the table, the colour version of the table, which is attached to the DNA report.
Can you just expand and go to Mole Katanga?
Okay. When it lets me. Oh, sorry.
Okay, so
So we are looking at the tabulated results of this sample, S004. This is Mole Katanga. And as you can see, there's a number relating to the specific area. And
okay.
this is taken directly from the EPG.
So basically, when you talk about, so if her DNA profile therefore is 16, 15, 17, 11, that's what you mean by a DNA profile, a series of numbers.
That's correct. A DNA profile is merely a sequence of numbers.
So if I say that I found Mole Katanga's DNA on this microphone, it simply means I'll get her
My
profile.
Lord,
What does it mean?
My Lord, yeah.
What does it mean? If it says that I found Mole Katanga's DNA profile on this microphone, what does it mean?
If all the DNA components were detected on that microphone matched that of Mrs. Katanga, she could not be eliminated as a contributor of DNA to that result.
Okay. My Lord, I hope I want to move to another area. I needed us to first understand.
So each, you can see that each area has numbers associated with it. If we look at sample 7, you can see that there are multiple numbers associated.
Yes.
In some of the samples, not all the numbers within this table, so sorry, from the EPG, not all the numbers on the EPG were carried over and appeared in this table, so there appeared to be some information missing.
Okay, we shall come to that in due course. But now using this table, and you had picked out sample 7. Yeah?
Yes.
We have just seen sample 4,
Yes.
which is a single DNA profile of Mole Katanga.
Correct.
And you have explained to us that where you see 2 pieces of information, it is one person. one piece came from the mother and one piece came from the father.
Yes.
Now my question to you on sample 7, for example, without going into what it is that at the second location, I am seeing 3 pieces of information.
That's correct.
What does that mean?
So that indicates that there is DNA from at least 2 individuals contributing to this result.
And why do you say that?
Because of the way DNA is inherited, a child will only carry 2 pieces of information in their DNA profile. If there are 3 pieces of information, it's a direct indication of DNA from more than one person.
Okay. Now, let's go back to where we ended last week. In the situation on yesterday but one. In a situation where, in the context of this case, Mole, Katanga, and Patricia are mother and daughter, how will that affect the interpretation of the DNA results in the context of this case?
I understand that Patricia is the biological offspring of Molly. Therefore, Patricia will share 50% of her DNA with Molly. It is still possible to carry out the initial interpretation of possible inclusion and exclusion. But in respect of possible inclusion, the statistical evaluation is far more complex, and very specialist software must be used to compensate for the relatedness between the 2 individuals.
Okay, I want to go to just very briefly to one other technical area, and then I want to come back again. And it's really within the same. If you go back to your report, you had talked about DNA profiling.
Yes.
And I just want you to explain to my Lord, I think it is on the previous page. I just want you, that would be page 30, is it?
Yes.
I just need to be clear.
That
Page 30. I just want you, we've now understood DNA profiling. We've understood it's simply a collection of numbers. And once I find these numbers, and I check whether these same numbers are somewhere else, and I say I can't exclude you. We've got that.
Yes.
The process of checking for the DNA. I think it's also captured in there. I just want you to very briefly explain how you test. You take a swab
Yes.
from a gun
Yes.
or from Mole Katanga. We've seen all the processes that can lead to contamination along that area.
Yes.
Finally, it gets to the lab man. What does the lab man do?
So the DNA profiling process is a series of stages. The first stage is to remove the DNA from the sample. The next stage is to measure how much DNA is present in the sample. The next stage is where we copy the DNA, so that's known as amplification. Once the DNA has been copied, the next stage is to separate out the individual fragments of DNA by size. And finally, it's the transition of the raw data from the separation into the visual representation that is shown in the EPGs.
And what are EPGs again?
So they're electropherograms.
And have we seen them in this court?
Yes, so there, that is the graph that I've been showing with Molly's profile.
Now, final question on that technical part of it. Just for my mind to understand inside the lab. In this case, we have so many samples. So is each sample tested separately through that process?
No, it's standard practice for batches of samples to be processed together. The number of samples in each batch is decided by each individual laboratory. The robots used can generally process up to 96 samples in one batch.
Now,
My lord, the last part. We got that they have been tested together. Each lab determines the samples, but the last one.
Okay, 96.
The robots which are used can process up to 96 samples in each batch.
they can process up to 96 samples in
The
each batch.
robots that they use are able to process, but I don't know whether that has been implemented into the Uganda police system.
Now, very briefly, you had talked about the letter on ISO certification.
Yes,
Do you have it? Do you have that letter in the documents on the?
oh, in relation to the laboratory?
Yes.
Yes. So the team appreciated the level of investments by government in the state-of-the-art laboratory equipment, highly trained human resources, information management system, and the ongoing implementation of laboratory quality management system, with intent to getting international accreditation to ISO 17025, ISO 21043, and ISO 45001 for laboratory testing, forensic science, and occupational health and safety, respectively.
Okay, then.
It is in this regard that the Ministry of Health adds that Uganda Police Forensic UPF Laboratory at Nuguru to the database of gazetted, certified facilities for DNA relationship testing.
And
However,
what is DNA relationship testing?
so that is a very different system to crime stain DNA profiling. Forensic DNA relationship testing purely profiles known samples from individuals, such as that taken from Molly KATANGA,
To confirm if what?
the paternity of a nominated child.
Okay. And so from this letter, you were telling my lord, what were you telling my lord about the ISO certification of this lab?
So whilst the laboratory are clearly working towards gaining the ISO accreditation, they as yet do not have that accreditation.
And that's according to this letter.
Yes, purely from this letter.
What about yourself? Are you supposed to be ISO certified?
All the laboratories which carry out the work in the United Kingdom are 17025 accredited. Our forensic science regulator published a set of codes which stipulate that any primary examination must be now carried out under ISO 17025. In light of this, I do not carry out any primary examinations at all. The work I carry out comes under case review, and this has no place at the minute under ISO 17025.
Okay. Now I want us to go back to the lab process. So we've now seen all the processes
Yes.
in this case.
Yes.
I want us to very briefly now, we've seen what's supposed to happen. I want us very briefly to go to what actually happened according to your review. And maybe I'll start by asking you. You've told us about the how in this case were the DNA profiles extracted? Okay, maybe let me ask the question again. In this case, from your review of the documentation, what was done?
The only documentation I have are the EPGs and the paperwork created during the stage where the amount of DNA was present, quantification.
There are EPGs and
So the EPGs and paperwork
And what?
created during the measurement stage of how much DNA was present, the quantification stage.
Okay.
From those documents, I'm able to determine that batch sampling was in place.
You tell me what my lot?
That batch sampling. That batch sampling was in place.
Yes. So they processed batches of samples together.
Can you, from the electropherograms, briefly show my lord how you can do that?
Yes. So during the DNA profiling process, each of the individual stages, the samples are processed with unique identifiers, and those relating to the batch that was on the separation stage under the software is labelled a project. In this instance, this project was labelled B15D06112023. And I'll just scroll down to demonstrate that on the top of each EPG.
My lord, I beg your pardon. Just the number for that.
It's there, it's on everyone, that's what she's showing.
Yeah, can we first write it, my lord?
You can see it here.
At the very top of the page.
B15G06.
My lord, can we write where the witness has read?
So from the sample one, each EPG is printed, and you're looking at where it says project. So it's B15D06112023.
Would that represent the date 6th November 2023?
It might, how the individual labs give titles to individual projects is entirely up to them. As long as it's unique and this project is identifiable, then they designate it.
So show us the batch.
So I'll
It
just
moves from sample 001. You were telling us.
001. You can see same 002. If I show you the first few. Three.
Maybe go to the last one.
And then it's 4. So these are all the reference DNA profiles, and this batch number continues to there were 20, so it continues to sample 23. You can see that this is exactly the same project number.
Okay.
Then, equally within every batch that's processed through the DNA profiling system, a negative control is processed.
And we'll come to that. We'll come to that shortly. You had told us you could see the batch number, and then there's something else you could see apart from the.
So after the batch number, I then looked at the paperwork created during the quantification stage, the measurement of the amount of DNA. And here again, and it gives the indication of how the sample number stays with that sample through every single stage of the process. So this means the actual processing is completely transparent. And as you can see at the start is sample 001. And it continues to sample 23, and then followed by the negative control.
So I just need my Lord to just see it. So that is, what do you call that thing there?
This is the paperwork which is created during the quantification stage. This is an automatic printout by the computer.
And what page is that on the?
So on this document, which has 119 pages, the quantification information begins on page 55
Page?
with it continuing on to page 56 where it finishes.
Okay, now.
Oh, sorry, it goes on to 57.
Okay, now. Let's go back to your report. You have the reference samples and the exhibit samples together, and
Yes.
you've shown us they're in a batch.
Yes.
What is your comment about that?
This is extremely poor laboratory practice.
Just stop there, let's record that. This is what?
Extremely poor laboratory practice.
Why, Ms. Hindley, is this extremely poor laboratory practice?
By their very nature, a reference DNA profile would have very high levels of DNA present.
And by a reference profile, what do you mean?
So this will be a sample from a known individual.
So in this case, what samples are you talking about?
So these are the samples from Molly, Henry, Patricia, those samples which were labelled sample 001 to 005.
And those how are those samples collected? Generally?
Generally, these consist of swabs, tooth swabs taken from the cheek cells of a nominated individual.
It's taken from your mouth.
Yes.
counsel is leading the witness to tell us about general things that are not specific to what she analyzed. Because we need to be clear. Is she saying that these particular samples were taken from any person's mouth, or she's giving a general view of what she thinks should be the practice?
What I understood is that the samples were taken from Henry, Bully, and Patricia, labeled 1 to 5, and that looking at the reference DNA profile together with the sample,
The exhibits.
with the exhibits, with the poor laboratory practice.
Extremely poor
The
laboratory
last
practice.
section.
The last section.
So answer that question from Mr. Mwaganya, which is this?
Can we, Mr. Karamba?
No, because you raised an objection as to where these swabs were gotten from, or whether you're dealing with a general question or a question. So, my lord, I want to ask the witness the question.
The clarification we ask is from court. If we can always cease fire.
No, my lord, there's no need for a ruling. I'm going to ask the question he wants
My
clarified.
lord, it's okay, we shall get the transcription.
No, that's an attempt to adjourn. My lord, let me ask the question he wanted.
Mr. Karamba,
Much
the
obliged.
question was asked for clarification.
Yes.
There was no answer.
Okay.
So that will come in.
Yes. So the question you've had the question, where is the idea of swabbing the mouth? Do you get that from general casework or from the facts of this case?
Exactly now is the point.
That's the question you wanted.
Yes, yes. True, that is the.
That is it.
So the standard practice is to use the mouth swabs. However, when we have samples from a deceased, we commonly see muscle tissue.
That's not understood.
No, she has understood. Continue answer.
But within this case in particular, within the DNA report, the reference DNA profile sample 1 from Henry Katanga was a blood sample from the deceased.
Okay, so
Just hold on. The prosecution was asking, they seem to be asking clarification in a different code.
yes.
What do I ask? I think the prosecution seek clarification from the response, from the witness on the response.
My lord, didn't they have time to cross-examine?
They will have time to cross-examine, but it is important that we do
Altogether. Much obliged.
My lord, what we had last is that reference samples were picked from, and she mentioned Patricia and another person that I didn't get properly, and she was saying they were picked from the mouth. So that's where we had lost her.
Okay? Is that what you said?
Yes, so generally the reference DNA samples are recovered from the mouth, the cheek cells of an individual.
Okay.
And in this particular case, from the DNA report, they have specified that a blood sample from Henry Katanga was used as his reference DNA profile, which is not unusual from a deceased individual. And the reference DNA profile of Molly Katanga, sample 4, was obtained from a buccal swab, buccal being the specific cells from the cheek of an individual, and equally from Patricia, the buccal swab was taken.
And what about sample 5? Oh,
Sample 5 is from Patricia, and that's the buccal swab. They were used as the main reference DNA profiles.
okay. So Mole, it was the mouth.
Yes, Molly
Patricia?
and Patricia, the mouth, with Henry from a blood sample.
And you explained to my lord why putting these samples together with samples of exhibits, the other exhibit samples, why that is, according to you, extremely poor laboratory practice.
Particularly when the samples are coming in for cellular DNA, when the expectation is the level of DNA will be low. If you have processed the reference DNA profiles of persons of interest with samples expected to have very low levels of DNA. This introduces a significant risk of DNA from the reference samples being available during the DNA profiling process to enter the low-level expected samples, for example, from the firearm. And it would not be possible to determine whether that DNA was as a result of contamination or if it was a true representation of the sample.
And then maybe we can take a translation. I'm asking a question. So my understanding is a batch sample is run within the lab. You've told us how reference samples shouldn't be run together. So we have samples we are testing to get a result at the end of the process. How can we be sure that the results this process produces are accurate and robust?
So as with any scientific test, we must run controls to demonstrate that the process has worked as expected. It is standard in all DNA profiling that a negative control, which consists purely of the chemicals used in the process and no DNA, and equally a positive control, which is a known DNA profile, and these are processed on every batch.
So how do I know that? How do you know a known DNA profile?
The manufacturers of the kits used, so in this case Verifiler Plus, produce a synthetic DNA profile, and within that kit the designations of that profile are given, and every batch is checked against that profile to confirm that the whole process has worked as expected.
So let me just ask one general question, then we're going to the specifics in this case. We have now reached the stage where we are running the tests,
Yes.
and we obviously don't know the results of the samples. That's what we want to find out.
Yes.
So we put in two results that we know.
We know the DNA profile of the positive control.
And what do we know about the negative control?
The negative control should be blank. There should be no information in the negative control. That is the only sample where we can truly pick up contamination.
Okay. So one, we know the result,
Yes,
and the other, we know the result should be negative
correct.
because there's no DNA inside it.
That's right, it's purely blank chemicals.
And that helps us understand that the results we have produced are robust because it's given us answers that we know.
Correct.
Now, in the context of this case,
Yes.
did you receive information on that quality control?
Yes, so within the EPGs were the results of the negative control and the positive control.
So first tell my lord about the positive control. Did you receive the result? Did you know what to expect on the positive control from the information you were given?
No, there was no confirmation of the designations which should be present. So the most I could do was confirm that information was obtained at every area being tested.
Okay.
Every area.
There was no confirmation.
I had, that's right,
Positive control.
that's correct.
So you got control.
that a full single source DNA profile was obtained.
She's realized to say that that result was as expected. Okay, so Ms. Hingley, what about the negative control on the DNA report?
Like what? Which confirms that the result was as expected.
Thank you, Maria. So this was the EPG of the negative control.
And this no DNA is being tested here, right?
That is correct. This should be absolutely blank.
And what does the EPG tell you, Ms. Hingley?
The EPG I've been provided is a partial capture of the result. As you can see in this top area here, the scale on the y-axis is not suitable for me to observe the DNA coming through.
No, just go slow. So on the first,
So-
we have our negative control.
Yes,
We have the boxes of locations on the first part,
correct,
and we are seeing nothing there.
correct.
And what are you saying about that?
The fact that we are not seeing it is merely because these EPGs have been printed from the system with the system told to print to the top, to scale from the top of the highest peak to the thousands, where when we look at the other colours, the actual information is coming through much, much lower. So I would expect if the analyst had changed this scale, I would observe DNA.
Okay, but now go to the next one, because, okay, that's the first box, but the second box, what do you observe on the negative control?
So as you can see visually on the second one, there's clear indications of DNA within this sample.
How many people's DNA is in the sample that had no DNA at all?
If we consider this area here, as we've said, I'm only expecting 2 pieces of information. Visually, you can see 4. So within this negative control, in my opinion, is DNA from at least 2 people.
They tested a blank sample and got results of 2 people.
At least, yes.
What does that tell you about the process?
This is demonstrating that there is contamination in the process.
Okay, just hold on. In your report, how do you describe this contamination?
In my opinion, based on the records provided, the amount of DNA detected in the negative control is significant, and I would expect an investigation to take place to attempt to determine the source of this DNA before any samples relating to this control can be deemed suitable for interpretation. By international standards, this constitutes a gross contamination event.
What page is that of your report?
That is page 11.
So just go back up. Let's unpack that. Yeah.
Yes.
This contamination in the negative control,
Yes.
does it have, what if any impact does it have on the rest of the batch?
The true impact I'm unable to determine because I do not know at which stage this negative control entered the process. From the paperwork, it appears that it came in at the very beginning,
My Lord,
extraction.
is it proper for her to speculate after stating she cannot determine?
No, she's explaining.
And then she's No, she's not explaining. She's actually guessing, speculating on record. Unless I didn't understand her.
Yes,
But if what she stated is that she is unable to determine that she want to go ahead and speculate.
Based on the rest of the batch, she's not able to determine,
Yes.
or she does not know at what stage this negative control enters.
Very good.
Yes,
She comes from misleading her to speculate.
No, my lord, it's not speculation.
It says it appears.
There are different impacts at different. Would that mean that there are different impacts? Would that, Miss Hingley, would that mean that there are different impacts at different stages?
Yes, so from the,
Let us first hold on. She said that she does not know at what stage this negative control enters.
Yes.
Full stop.
Yes.
So did you ask another question?
Yes.
She had continued to
Are
say
there
it
different
appears.
impacts at different stages?
yes, there is. And the difficulty I have in confirming where is because the way the sample has been named on the quantification stage is NTC, which does not mirror the EPG. So it would be standard practice that the sample NTC would be the negative control.
What page is that which has NTC?
So it's some A4 here, after the batch sample 23.
No, what page is that on D5? On the document you have, what page is that?
This would indicate that it's at the quantification stage, and therefore the negative control relates to the extraction of the batch.
So in the event that it relates to the
Let us first still explaining something
yes,
relating to the difficulty she has in farming, and then you ask something. Right?
much obliged. Much obliged.
So there are different impacts at different stages.
Yes.
And then the difficulty, you are saying the difficulty I have in confirming. Let's finish.
Confirming?
In confirming whether the sample referred to as NTC on the quantification batch is actually the same as the sample labelled as negative control. So whilst I can't confirm,
Why I had stopped you before, I just need you to confirm to my lord what page.
My lord, can we finish that?
No, my lord, because there's a page number she's showing us with
Let's
NTC.
finish writing what she has said.
Much obliged.
The difficulty I have in confirming whether the sample referred to as NTC on the quantification batch is the same as the sample labeled as negative control.
yes, that's correct.
In the
So
electropherogram, my lord, is what she had added.
whilst I can't confirm which stage it's come into, therefore I can't inform the court the true impact.
Okay.
At the very least, the whole batch should have gone back through the process to be recopied to try to determine whether it entered at the amplification or copying stage. And if necessary, if it is confirmed at the extraction stage, every sample will need to be resampled.
Sample need to be resampled.
Resampled, so they'd need to go back to the original exhibit and take a new sample because it's demonstrating that the extraction itself has contamination.
So that means it is useless. But
Mr. Parama, let's finish. Please.
much obliged, my lord. I'm sorry. I'm sorry, my lord.
So irrespective where it came in, no,
It
it was
would be resampled from the original exhibit.
exact
Because it will demonstrate
contamination at the extraction stage, which is the first stage of the process. and therefore the only way to start again is to go back to the original exhibit.
So, this page here, which is showing, is what page number is that?
That in the page of the EPG document is page 56 of 119.
It's page 56. And what stage is this?
This is the printout from the quantification, so the stage where the amount of DNA is measured. You wouldn't put in a negative purely for quantification, so it would imply that this negative relates to the extraction batch.
It relates to the extraction batch.
Yes.
And if it relates to the extraction batch, we should disregard all the... What should we do to all the results that have come?
If, upon reamplification, the DNA detected still remains, the contamination could only have entered the process at extraction.
Okay.
Because there's no step prior to extraction, the only option is to resample the original exhibit, and all profiles relating to this extraction batch do not meet the criteria for evidential purposes.
And from what you have you seen any retesting?
No, there's no evidence of any retesting or any investigation into the source of this DNA. I would expect internal quality documents to be available, an investigation launched, and equally evidence of a contamination log within the laboratory.
Now, I just want you to confirm this negative control that you've seen
Yes.
in relation to what impact does it have in relation to exhibits from the gun?
Currently, in the absence of any retesting, the profiles relating to the gun do not meet the criteria for evidence, and currently they should be disregarded.
Now, you're talking about profiles. I'm asking you about the results from the DNA profiles on the gun. Or is that the same?
It's the same thing, yeah. So the DNA profiling results, which are contained within the DNA report, are currently not useful and should be not considered in the context of this case until an investigation into where that contamination has originated from is conducted. Without a full investigation into the source of that DNA and how that DNA entered the process.
Okay. Okay, now, my next area, I'll go to it very quickly. Apart from that incident of apart from that gross contamination event, what other comment do you have about the process?
So the final...
My Lord, just to be sure, the witness said contamination or gross contamination?
Gross contamination event.
Witness said gross contamination event?
Yes.
In my report, I described this contamination as being significant.
No, my Lord, there's where she says the event, this constitutes a gross contamination event by international standards. I have that recorded there.
My Lord, we can save on that type, we can verify from the transcription.
Okay, now I can just ask it again. What does your report say about the existence of the negative control? What does your report say about the existence
No, my
of
lord, it is not proper for him to ask the same question after he has proposed an answer to the witness. Since we have the transcription.
No, my lord.
And the record, and the court may or may not have captured it. We shall rely on the transcription. Because we had
My lord, can I be allowed to continue my examination in chief?
May I be heard by court, please? We had another thing, my lord. Counsel put another thing. We made an inquiry. It doesn't appear to be on the typed record of court. And so counsel, after proposing an answer, cannot put the same question.
I'm not proposing an answer, my lord. I take exception.
You have said so.
I take exception.
Let us proceed.
Yeah,
My lord, I take exception.
Let us proceed.
Mm.
How did you describe?
So I was
The record is there, if it is something different, we will see.
Yeah.
I was reading from my report on page 11 of my report. After I've said that any samples relating to this control must be reworked before they can be deemed suitable for interpretation. And the final line is, by international standards, this constitutes a gross contamination effect.
Okay. Now, my next question. We have seen poor laboratory practice and one gross contamination event. I want you to explain to my lord what, if any, other events of contamination you saw.
So the final stage of the DNA is the separation of the individual fragments of the DNA. In order for these fragments to be appropriately sized within the batch, a sample which is generically referred to as an allelic ladder, and this contains the most common pieces of DNA observed at each of the areas targeted. So on page 13 of my report, this is what an allelic ladder looks like. Obviously, from the previous EPGs, we're only seeing one or two pieces of information. The allelic ladder contains the most common at every single area. Hence, there is far more information in the allelic ladder.
So just hold on. My lord, I don't know if we're together.
Yes.
So what you're telling us, a normal DNA profile would have two pieces of information at every location.
For a single profile, yes.
For a single profile.
Yes.
The ladder would have pieces of information at every location.
At every location and multiple pieces of information at each.
You'll have multiple pieces of information.
Yes.
So in the context of this case, what has the allelic ladder got to do with our case and the samples that are in this case?
So during the review of the samples,
Maybe let's use the actual samples. You go to the electropherogram.
and this is entirely as I would expect had the ladder been introduced to this sample.
Go slow. Sample 23.
So sample 23
What page is it at?
on page 50.
There is a clear.
So sample 23 on page 50. Again, the EPG is a partial capture because of the scaling. So I've not got enough information in this one. However, as soon as you move through, that in my opinion is characteristic of the ladder contaminating that sample.
So that is because at every what do you see at every location?
At every location you have multiple pieces of information. That in itself does not mean it's ladder. It's the characteristic strength of the relative pieces of information that, in my opinion, make it characteristic of the ladder.
Now, what was sample 23?
Sample 23 was a cartridge casing recovered from the bed sheet, Exhibit 7.
And what did analyst number 8 say he had found on cartridge number 23? On sample 23.
From the DNA report, analyst 8 stated that a partial mixed DNA profile of at least 2 donor contributors was obtained from this sample.
Now go back to the sample. Now, when you find this type of result, a ladder, what does it mean?
It means that this sample cannot be interpreted at all.
Now, how do you describe the presence of the ladder in the sample in your report?
In my report, the EPG relating to this sample showing far more complex results that has been reported, and I therefore disagree with their interpretation.
Okay.
The representation of a large number of DNA fragments in the result, in my opinion, indicates that this sample has been contaminated by at least the allelic ladder, such that it is not scientifically possible to carry out any meaningful comparisons. And by international standards, this constitutes a gross contamination event, which in my opinion renders the result unreliable.
Now, I just want to understand, is this just the result of the cartridge that would be unreliable?
No, again, an investigation needs to be launched as to how that ladder came to be in that sample.
Okay. And if it came from, say, the extraction stage?
The ladder is only introduced into the DNA profiling process at the separation stage. If it is found in stages prior to the separation stage, because of the way a laboratory must be set up with airflow running from the start to the finish process, it should not be possible for DNA later in the stage to be found earlier in the stage. As such, if ladder is found earlier in the stage, the laboratory must stop processing samples, carry out an intensive deep clean, run samples or blank samples known as environmentals to demonstrate that the laboratory is clean.
Just let me put it together. Together. You say it must stop running samples. Must it stop running samples in this case, or must it?
They must stop completely. If you are a DNA processing laboratory and you have DNA which is from post-amplification in the system before amplification, the effect of contamination is so gross that it renders all samples unreliable, hence they must stop processing if something like the ladder is found in an earlier process.
What is the overall impact of that level of contamination that you have described to my lord on the DNA results in this case?
In my opinion, in the absence of any investigations, rework on all the samples, none of the DNA samples are fit for evidential purposes and should be disregarded.
Well, I'm happy to take a translation.
Oh. So it was also present in sample 29.
What was sample 29?
Sample 29 related to item 24.1, which was an adjustable walking stick. It was equally present in sample 30,
What was sample 30?
which was item 24.2, a wooden stick.
Yeah, we call them in Kony.
These two being on a different batch to that previously seen on item 7.
So in a subsequent batch you found
Yes,
the ladder.
yes, the samples 29 and 30 were on subsequent batches.
And just briefly, we'll look at that at our own time. Did you also find contamination in the negative control in other batches?
Yes, there was.
So now in the totality of the case, not just on the batch that has the gun, but in the totality of the case, what is the impact of this on the results?
It is apparent that the actual DNA profiling process, irrespective of the batch, has gross contamination. And without further intensive investigation, none of the DNA results should be presented before court.
Okay. I want to move to my second last area. I'm being told how to slow down, my lord. But Miss Hingley, Miss Hingley,
Yes.
nonetheless, you've taken us through recovery of the evidence and the challenges, the laboratory practice. Nonetheless, we have DNA results.
Yes, there are.
And I want us now in this next segment to briefly go over the actual results that we have.
Okay.
And notwithstanding all the issues you've raised, we have DNA results attributing major and minor DNA profiles on certain samples.
Yes, that's correct.
I just want you to tell my lord how the conclusion, we have the conclusion and the calculations to attribute major and minor DNA profiles are arrived at, very briefly. And if we could use your report, maybe it could be faster.
Which sample should I?
No, generally, I just
Generally.
want to understand.
So, when we review DNA profiling results, the first thing is a general overview of the number of contributors to a result. And then following that, visually, is it possible to separate out the pieces of DNA into individual DNA profiles? So in this instance, just using sample 20 as an example, which is page 44 on the EPGs, this is a major profile and a minor profile. This result has much stronger pieces of information and much lower pieces of information.
No, Ms. Hingley, maybe using the EPG, I just need to understand what you say. Where is a major and where is a minor there?
So the graph is showing the height of an individual piece of information. So here, if we look at this piece of information, the height is 10,000 high, where this piece of information is only 784. DNA adheres to specific rules when it is well amplified, and therefore it is not possible at this area for the profile to be anything different from the designations 11 and 16, the big peaks. A true major DNA profile, you can guide your eye along and literally pick out the strongest, the tallest peaks, and they will all form one DNA profile. So no mathematics is required with results such as this.
That's page 44.
That's page 44, sample 20.
And sample 20 is
The barrel,
the barrel.
yes.
And just for the record, whose DNA was more predominant on the barrel?
So the major profile, which you can see, is Henry KATANGA. And you can, unlike Molly, you can see here on the Y, the male specific, we now have a peak.
Okay, so with that sort of profile, no mathematics required.
No mathematics is required. Depending on the country, we all do different things, but in the United Kingdom, a major profile, full profile, is automatically given a likelihood ratio of 1 billion.
Okay. So when you're reporting this profile, you'll say it is 1 billion times.
Billion times more likely that the DNA originated from Henry KATANGA rather than from an unknown, unrelated individual.
Okay. And is that the same language used in this case, or it's a different language?
They have performed calculations, formal statistical calculations on every single result, so the terminology is subtly different.
What word is that, subtly?
It's subtly, it's indicating the same thing, but because they have formally done a statistical calculation, the terminology is subtly different, but the end stage is it's 1 billion times more likely.
Okay. So, when a statistical calculation is done, what does that entail?
Firstly, the analyst must review the DNA profiling result and determine what they deem suitable within that result for the statistical evaluation. That data is then entered into specialist software. In this instance, the laboratory used the software Euroformix. It's incredibly powerful software, and it uses the information within the profile, such as peak heights, to determine the frequency of each of the pieces of information with every possible combination, and then creates a final likelihood ratio of the significance of the nominated individual's DNA being present.
Okay, now I want you to very briefly go to your report and tell us what, if any, comments you make about the software that was relied upon.
Just go back up. There are multiple softwares available to forensic laboratories for the interpretation of mixtures. There are different systems. There's software known as open source, which means it is open on the internet for anybody to download.
So that is free software that I can download from the internet.
Yes.
What is Euroformix one of those, or it's another type?
Yes, Euroformix is an open source. Now that in itself isn't an issue.
Before you even go to an issue, what you're telling me is that the Uganda Police Laboratory downloaded its software from the internet. Is that?
That is inviting the witness, sure. Yes.
I'm disagreeing. No, it's open source. Is that the meaning of open source? Does open source mean that I download my software directly from the internet, or does it mean something
My
else?
Lord, the best that he can ask, what does open source mean?
What does open
But
source
a
mean?
lawyer in evidence.
Well, there's no need for lengthy objection. I'm conceding. What does open source mean?
And the problem, my Lord, even when we are making a contribution, Mr. Karohanga doesn't listen.
My
My
Lord,
Lord, it is
My Lord, I'm surprised that my colleague is objecting to my agreeing with him. What does open source mean?
What Mr. Kapanga
Behave.
doing exactly that.
I'm being behaved, my Lord. Today I'm on my best behavior. I'm even agreeing with
This
Mr.
is
Morgan.
not just being
I am much obliged, my Lord.
allow the witness to answer.
Okay.
Suggestions.
What does open source mean?
So yes, open source means that it is open to anyone on the internet. And it must be appreciated that all the techniques we do use in forensics are not purely used in forensics. There's an awful lot of research laboratories that again use the tools. So in this instance, Euroformix is an open source program.
Okay? Now, the impact on our case. Or maybe the explanation you are giving, my Lord, about software.
That's not the
In order to be used in the forensic context and to meet the evidential level required, the producers of the software must demonstrate that the software and the mathematics within the software work as expected. It is then down to the laboratory
The owners of the software must demonstrate
that the process and the mathematics within the software work as expected. The next stage is the responsibility of each individual laboratory, and they themselves must fully validate the system for the samples and their processing procedures. This can only be done using known DNA profiles, as we never know the ground truth of any crime stain submitted for DNA profiling.
Beg your pardon, as we never know.
The ground truth, so exactly what was in that sample. It is also essential that for use in forensics, that the laboratory lock down the software so no one can alter any of the parameters within the laboratory. In the process of my review, I requested the validation documentation. However, this was not provided, and I can therefore not confirm how this system is being used.
So, Ms. Hingley, in the absence of the validation protocol, what comment do you have about how the system is used in relation to the results in this case?
I cannot confirm whether it's reliable or not. If they have fully validated it and can demonstrate that the figures being produced are robust, then the software is validated for use. However, in the absence of that, I cannot confirm. And the added complication in this case is the fact we are dealing with close biological relatives. And I can see no evidence that the software at the time of its use was able to robustly compensate for close relatives.
Okay. So, Ms. Hingley, again, notwithstanding that we are unable to verify that there is contamination, that there is poor laboratory practice, nonetheless we have results. I want us to go to two very specific results. I want us to go to the result on the trigger and trigger housing. You had of the secondary swab, the one which was taken after the trigger was swabbed. You had explained to my lord that the first swab would have removed the DNA from the trigger, but that secondary swab, I want us to go to it, and I'm going to be very brief and quick on this one. Firstly, if we can just go to the electropherogram itself. So what sample was the trigger and trigger housing? What page is that?
This is page 42 and it's sample 19.
Okay. Now, I'm first going to ask you about a very specific result, and then I'm going to ask you very general questions. Do you see the first location there?
Yes, the same as all the other samples, D3.
Do you see the first result?
Yes.
What is it?
Labelled within this EPG, we have pieces of information designated 14, 15, 16, and 17.
Now I want you to look at 14 and just tell me whose DNA was 14. And I'm looking specifically, and maybe we just stick at the electropherograms, maybe it'll be easier. If you can take us to the sample of Henry Katanga. Did Henry Katanga give us a 14, then the sample of Mole Katanga, and the sample of Patricia?
Has 14.
He has 14.
So Henry has it.
So Henry has it. What I want you to do on the first area is to go back to the magazine, and maybe use the table.
The table.
Yes, and just confirm for us. No, I want you to go to the electropherogram
Of 19.
of 19. Yeah? And then I want you to confirm if the results at location 1, if they are shared by Henry, Molly, and Patricia, just so we can understand what we are talking about. So Henry has, if you go to sample 1, Henry has 14 and 15, so we can
Yes.
see them.
Molly has 2 copies of 16,
Yes.
and Patricia has a 15 and a 16.
So now look at result 17.
So this result here
Yes.
has not been carried through. However, I have no
First
interpretation.
listen to my question. My question is, result number 17,
Yes.
does it belong to Henry, Molly, or Patricia?
No, it doesn't.
Thank you. Just wait for the next question. We'll come to the impact, but before you go to the impact, I want you to go to, you have told us about the Euroformics calculation.
Yes.
I want you to go to the Euroformics calculation in this case. So we have the allele number 17 at location, what's the name of the location?
D3.
So location D3 has what allele?
14, 15, 16, 17.
And 17 belongs to?
Currently unknown.
17 belongs to unknown. Now go to the DNA report and let's see how the statistics were calculated.
Sorry. Sorry, I've skipped it.
Isn't that the one?
That's 17. This is 19.
Okay, now we want to come to how the statistics were calculated, but this is on the DNA report.
Yes.
There are no
Page
page numbers.
114.
Maybe that's one. Okay. Now, Ms. Hingley, let's first go up before you go to how the statistics are calculated. Explain to us the first paragraph of what we are seeing. Just read it and tell us what that means.
So at the very top,
Yes.
it's confirming that the report was generated from Euroformics version 3.2.0.
Okay. And who is the user?
The user is recorded as L. Mutesi.
In forensic science, would that mean that's the person who entered this onto the system, or can someone else use their credentials?
Is that a matter of science or a matter of fact?
Okay, let me rephrase the question. Is it a matter of science to know who the user is?
Yes, part of the ISO 17025 accreditation is complete transparency in the whole process, which means if any sample is audited, it can be determined who carried out the work, when it was carried out, and if necessary, who checked the work. So it should not be possible for someone else to use someone else's login onto the system.
Okay, and does this show you who carried it out and when it was carried out?
From the information I have, the user who carried it out was El Mutesi, and this was carried out on the 9th of November 2023 at 16:53 p.m.
Okay, now, I want you to show us where in this calculation can we find the alleles that we have just seen on the electrophoregram.
So if you go down to page 115, under frequency data,
Okay?
So what part of the DNA report is that?
It's the back, my lord. It's the last annexure. The one, yeah, it's the. I think it's annexure 4, is it? It's the last annexure. There are a series of samples. We're looking at sample 19. So you are showing us,
so this is page 115,
okay?
and under frequency data, you can see that under this area D3, the pieces of information which have been imported into the software are 14, 15 and 16.
Just stop there. Our mystery 17, has it been inputted into the calculation?
It has not, but I don't know why.
Okay. That is sufficient for our purposes. It hasn't, and you don't know why. It's there. Maybe explain it again. You're saying that on D3, what samples have been entered in?
So D3, what this is showing are the individual pieces of information inputted into the statistical software. And then after that, it is saying the frequency of that piece of information. So here we have designation 14 with its frequency, designation 15 with its frequency, and designation 16 with its frequency. There's no evidence that the 17 has entered the system.
And this thing we are looking at, how do we know this is sample 19? Again, sorry?
So recorded at the start, underneath where it says the user is the data, and it's saying the profiling kit verifier plus, it's stating the population it's considering for the frequencies, Uganda, and here under evidence, so on page 114, under evidence is the identifier of the suspect in question. So this is sample 19.
Okay, now, I want us now to go back to your report. In spite of what we have seen and what we can't explain, there is a conclusion, according to the DNA expert, that it is likely that Mole Katanga contributed more DNA than Henry and Patricia by 1 billion times, and that she's a major profile. Do you agree, and can you explain your reasons for agreeing or otherwise?
No, I don't agree that there was a major profile present. When reviewing the profile, in my opinion, there were major DNA components from at least 2 individuals that could be accounted for by the presence of DNA from Henry Katanga and Molly Katanga. Therefore, they could both be considered as substantial contributors of DNA to this result. The components in the reference DNA profile of Patricia were fully represented in this result, such that she could be a low-level contributor of DNA.
Okay. What impact does the fact that Patricia and Mole are mother and daughter have on the results of this sample?
So as previously stated, the fact we're unable to eliminate both mother and daughter creates an added level of complication that there will be 50% shared between the two, and only a very specialised statistical software is able to compensate for that and provide accurate representations from the nominated individuals.
Okay, then the language used, the report says that according to them, I see it says those two propositions in the report. What,
Yes.
if any, comment do you have on that?
The calculation has been processed on the assumption that DNA from Henry is present. It is then stipulating that all individuals are unrelated to each other. Therefore, this statistic is not representative of the DNA profiling results, including information we have about contact Patricia had with the firearm, and therefore the statistical evaluation presented should be disregarded in relation to this sample.
You also talk about the wording of the propositions. Could you explain to my lord what you mean?
Yes, so the actual propositions are just considering the presence of DNA from Molly Katanga, yet the final conclusion, the term major contributor has entered. That is not actually what the statistic is showing. The statistic, if deemed appropriate, is merely 1 billion times more likely that DNA from Molly Katanga was present rather than from an unknown, unrelated individual. The statistical software does not dictate whether she was a major profile or not.
Yes. And so this sample 19, what sample was it?
This was from the trigger and trigger housing.
I want us now to very quickly and briefly go to the magazine. But maybe before that, if we could just go back to the Euroformics results.
Yes.
Because you said that there's a 43%. I want us to go to the Euroformics and you show us those results.
Of the Euroformics or the EPG?
The Euroformics. So where are you getting 43% from?
Oh, yeah. This is it. So the two propositions the software considers. One we term HP, which is the prosecution hypothesis, and the latter being HD, which is the defence alternative. So from the prosecution hypothesis, the software has estimated that C1...
You can show us with the mouse.
Oh, I'm sorry. So estimates under HP, this is page 114 at the bottom. The proportion of C1, who is Henry, is 0.38, 38%. The proportion of C2, Molly Katanga, is 0.43 or 43%. C3, Patricia, 0.18 or 18%.
So just next to them there are numbers. What are those numbers?
I understand that this is an error rate incorporated into the software.
So that's the error rate for each.
For each one, so whilst it's been reported as 38%, the producers of the software have equally considered that there is an error rate in that percentage.
Okay, now I want us to go to the magazine. About to wind up, I just have 2 quick areas and then I wind up. And I want us to go to the electropherogram on the magazine.
My lord, you are sorry, my lord. You need to write something where she went, or where there was C2, C3, C2, my lord.
It is here.
So under the expectations under HP, the mixture proportion of C1 is Henry, C2 is Molly, and C3 is Patricia.
Okay? If we could just quickly go to the electropherogram for the magazine. You'll tell us what page of D5 it is.
So the magazine is sample 17 and the EPGs is page 38.
Okay?
So again, I've got a partial capture of information due to the sizing of the axis, but where I can see detailed information, particularly FGA being a good example here.
Yes, I want us to go to FGA. And before, again, I want us to do the same thing. There's something marked there at 25,
Yes,
a little 25.
yes.
Does it belong to Henry, Molly, or Patricia?
No, that is DNA that doesn't match anyone. Oops, sorry, just put it on. Oh, I can't do that. No, so that 25 doesn't match anybody.
Maybe just show it to us on the electropherogram so we're all on the same page.
Sorry?
Oh, we have, okay, so 25 doesn't belong to anyone?
No.
Maybe if you show us the color table.
1 second. Sample 17. FGA. There. Can everyone see?
Yeah, you can see a bit.
The colour doesn't help.
22, 23, 27. Yes.
So it's this area here. This is FGA.
Okay. And so we have a 25.
No, 22.
So if I go back to the EPG,
Yes. Even if it is easier to just use the EPGs, maybe we can do that.
Okay.
We compare. We know that at location FGA, we have 25.
the designations labelled 22, 23, 25 and 27,
Okay.
all of which have been carried across to the table.
Yeah, yes.
Oh, no, 17. 22, 23, 25 and 27. So all the information has been carried across in this instance.
To the magazine.
Yes.
Now I'm asking the 25. Does Henry have the 25?
At FGA, Henry has 24, 27.
What about Molly?
Molly has 22, 23.
What about Patricia?
And Patricia has 23, 27. So none of the nominated individuals have the 25.
Go back to D12. On D12, I want you to go back to the electrophoregram. I want you to go to D12.
So this, you can see that there are 3 labels, a 15, a 17 and an 18.
And are they relatively tall? Is it a substantial amount of DNA provided at that location?
The DNA has met the requirements set by the Ugandan police for being DNA.
What location is that?
D12. What page is that on the electrophoregram?
That is page 39.
I want you just to look at alleles 17 and 18, and I want you to tell me if they belong to Henry, Molly, and Patricia.
No, again, none of them have 17 and 18, so this is indicating DNA from an unknown.
From an unknown person.
Yes.
Cindy, before I even come to the issue of major and minor, if it's a conclusion that it is a billion times more likely that Henry, Molly, and Patricia contributed other than an unknown individual, what would you comment about the accuracy of such a conclusion?
In my opinion, I agree with the initial interpretation that it's not possible to eliminate Henry, Molly, or Patricia. On reviewing the DNA profile, if they are all contributors, then as page 20 of my statement indicates, there are clear indications of DNA from a further individual. Therefore, the statistical evaluation of assuming 3 contributors is not a reflection of the actual result. And again, the statistical calculation carried out is misleading and needs to be redone.
Now, if we go back to the electrophoregram, according to the Euroformics, Molly contributes 67% and she's a major. Do you agree that there is a major profile here and that it belongs to Molly Katanga?
Can I double check that figure, please? So the statistical printout for sample 17 begins on page 111.
And just take us through the same thing, just go back up and just take us through the same thing again.
So as before, confirming the report has been generated using the software Euroformics version 3.2.0.
And the user is who?
The user is the same, El Matesi,
Okay.
and again created on the 9th of November 2023 at 16:46. In this instance, again, estimates under HP. We have C1 is consistently Henry, which is 0.26. C2, Molly, 0.67. And 3 is Patricia at 0.06 or 6%.
Do you agree with that statistical evaluation?
In my opinion, visually, I would not be able to determine a major profile from this result. And equally, in my opinion, the number of contributors the system has been told to consider is inaccurate and does not represent the actual DNA profile. Therefore, those percentages should currently be disregarded until a robust statistic is performed on this sample.
But visually, who is the major profile here visually?
It is not possible to determine a major if just visually looking at it, this 25 at FGA does not match any of the nominated individuals. And this piece of information is roughly similar to everything else. So there is no major profile in that result.
What did you say what would you comment about the D12? D12, if you go to the same do the same exercise on D12.
D12, we need to be very careful because, in my opinion, we are starting to lose information from the result, and therefore we're not seeing everything there.
Explain.
When DNA starts to become limited, we don't always capture all the information at all the areas, and this is known as a partial profile. So in this instance, where I had previously referred to as DNA adhering to rules, that only applies with samples of good quality DNA. Once the level of DNA starts to drop, we're more free. The DNA doesn't adhere to the rules. And in this instance, we have a very strong 15, but in a 4-person mixture, you could have all of them having that piece of information and therefore inflating it. So there is no major profile in this result.
So you're saying that all Henry, Molly, and Patricia all have 15. Is that what it is?
Let me double check. Yes, so Henry 1520, Molly 1519, Patricia 1515. So if these three are contributors to this result, then those three alone, there are 4 copies of the 15. So it's artificially inflating the height purely because it's a mixed DNA profile.
So go back to the electrophoregram and you see. So our mystery donor contributed all the rest of the DNA there.
I don't know. The result is showing at least 4. The true number of contributors can never be determined. We use how the profile is showing itself. So this is at least 4 contributors.
Okay.
And if Molly, Henry and Patricia are contributors, then this single piece of information, this 15, is actually 4 copies of 15 on top of each other. So individual profile wise, I'd expect it to be relatively the same in height as the 17 and 18. So again, no major profile.
Okay, now, essentially I want to wind up this examination in chief with just 2 questions. And they are general questions.
Before we go there, I have stopped recording at when she said when the quality of DNA starts to grow and the DNA does not adhere to the rules.
Yes. We're far more cautious with allowances of relative proportions, how the peaks react to each other. We accept far more.
Owing to the storage, recovery of items, the handling, the submission to the lab, and the handling of the item in the lab, what impact would that process have on the DNA results in this case?
My Lord, is he asking about the handling of the items in the lab in this matter, or generally? Is he inviting this witness to give an opinion as to how items were handled in the lab in this matter?
Yes. Yes. Yes, based on your report.
So,
Her report. Yes.
yes, my Lord, as to the handling of items in the lab.
In this case,
That there should have been a log.
This.
That's what you
yes. Yes, so the handling, storage, recovery, submission to the lab, and the handling within the lab, according to your observations in the report, what impact would the entire process have on the validity of the results before my Lord?
In my opinion, in their current state, the DNA findings in relation to this case should be disregarded. They are not scientifically sound.
Okay. Nonetheless, Ms. Hingley, in the context of DNA, if we were not to disregard the findings, what impact would the alleged DNA, if believed, what impact does it have on the single question before this court? If believed, can that DNA be used to attribute? Can that DNA be, what conclusions will that DNA evidence have on the allegation that Mole Katanga shot and killed Henry Katanga?
My Lord, I do not remember that is the question before this court.
I'll rephrase, my Lord.
My Lord,
My Lord, let me rephrase.
does
My Lord, let me
agree with
rephrase.
us that that is not a question?
I don't agree with you, but I'm going to rephrase. My Lord, allow me to rephrase.
Mr. Barrow.
Allow me to rephrase, my Lord. In the context of the cellular DNA in this case, because we don't have body fluids, whether or not it is disregarded, it should be disregarded, but supposing it is not disregarded, in the context of the cellular DNA in this case, what can you tell us about the ability of that DNA to determine whether or not Mole Katanga shot Henry Katanga?
My Lord, that has never been a question. We do not even remember that the DNA expert had addressed that question. When did that ever arise?
Okay, handled the
That
gun.
is a misperception of the scientific input of this DNA.
I don't know if that's a knowledge prosecute.
No.
That is the only question that this court is answering.
It's a misperception.
Mr. Karam, are you trying to determine if the results,
She said it should be disregarded.
if the results were to be regarded?
What impact do they have on the context
Yes.
of this case? On the context of this case, Uganda versus Mole Katanga, where the accused is accused of having shot her husband.
In my opinion, the DNA findings do not help to address how the DNA got there, who put it there, when it was there, and therefore cannot address the issue of who fired the gun.
So the DNA,
The DNA findings
findings
do
don't
not
address the
help.
question of
How the DNA got there, who put the DNA there, when the DNA was deposited, and therefore the DNA results cannot help to address the issue of whether or not Molly Katanga fired the pistol.
My Lord, I am done, and I'm happy to hand over the witness to the very kind and able hands of my learned friends.
missing, so the results will not even point to whether Mr. Katanga shot himself.
The DNA results do not help address this case, the pertinent question of who fired that gun at all. That they are meaningless in the context of this case.
My Lord, maybe just one last question. Sorry. And Ms. Hingley, if we were to find out who fired that weapon, what scientific study should we have looked for?
The only way you could demonstrate contact with that firearm, obviously the contact doesn't necessarily mean they were the individual that fired it, but the only thing that would help contact is fingerprints. DNA would never address how that DNA got there.
Now, my Lord, I am truly done. I'm ready. Finally, my lord, I'm ready to hand over the witness to the very kind hands of my learned friends.
My lord, as noted on this amendment, we requested for the type of transcript for us to be able to follow, because there are some areas where we're not able to capture them in our notes. We were given a transcript and we saw it this morning. However, we also request for this one as well to enable us conduct our cross-examination. But also, we are requesting if the witness can be kind to bring us a copy of the letter that gave her instructions to do this analysis.
I'm sorry, my lord. There is one last question. I just beg your indulgence. My lord, I pray.
Can't you be organized?
My lord, I just pray for one question.
Last question.
Last question. One last question, my lord.
So now what do we
The colleague is talking.
No, please. No.
You can't assume that these matters are more urgent.
is to wait until I finish addressing what, then we can jump in.
My apologies.
So sit down and relax. We'll come with your question.
Okay, I want to thank my senior colleague for her very gracious advice.
So, my lord, we will need the letter of instructions and then also a copy of the transcription for us to be able to do the cross-examination. From the one that we have seen, it's already available. Yeah, hold on, 141. We could use tomorrow to review.
But which two computers are
But my lord, before we do the dates, can I just ask my last question? I seek your indulgence.
No, but afternoon.
My lord, just the question. One question. One question to the witness.
This is your last question.
It is, my lord. So you have seen your report, right?
Yes.
And you identify that it's signed on every page, right?
Yes.
My lord, I pray to tender it in as a defense exhibit.
The prayer is to tender the report of the witness. You have, the prosecution has no objection.
Much obliged, my lord.
No, my lord, we are not objecting to him identifying. Did we understand the question
Yes.
that is asking her to confirm
She
having
has
signed?
confirmed and now he is
application
praying
is
to have the witness tender.
My lord,
her
may we look
report.
at the report?
Someone that that is in Mr. Farham you have agreed to what you are not had
No. This Akarwanga's interjections.
I was actually shocked that he had agreed without the process finally.
But you have to say something.
Separation of evidence had started to die down.
Report of the witness is marked DE 29.
Much obliged.
It's 29, isn't it?
Indeed.
Yes. What is the problem?
I don't know what's wrong with it.
Someone wants to create an opportunity for another.
You know? I can't believe it.
No, I was just being sure that my colleague is in agreement.
Yes.
My Lord, my understanding is that we had it tomorrow. And my colleague is saying tomorrow they will proceed and they need time. What are they saying? That they don't have time
They're
tomorrow?
saying that tomorrow they need to interact with the testimony of the witness.
I think they need it tomorrow.
Is that what?
But my Lord, the testimony is that same report, which my colleagues have had. You remember, my Lord, when we got these dates from you, we indicated that we had a witness who had traveled here and gone back without testifying. Then you gave us these 3 days so that then she can fly in here, she can testify, get cross-examined, and then leave. Now, my colleagues are saying that they are not even available tomorrow to finish. They are not even available next week. Except if, my Lord, if we are getting a date and the witness can be cross-examined on Zoom. Because, my Lord, you have somebody who's been in prison for 2.5 years without working. How are you supposed to support witnesses who are traveling and you are spending colossal sums? So why not the DPP is dilly-dallying and finding time when they are available, when they are not? This court must be alive to the livelihoods of people. We have days, let's use them.
And also the fact that this report was disclosed.
And if we don't have them, this report that the witness has limited herself to was disclosed 2 months ago. It's not a Bible. They knew she was coming. They were even ready to ask for her work permit. She came here, they looked at her passport. She flew in here, my Lord, and we informed you. So, my Lord, I need you to understand. Yes, ma'am. Yes,
I
ma'am.
understand that the witness has flown in. I understand all that you're saying.
Yes, my Lord.
But the prosecution has put before this court the fact that they need to interact with the report on the basis of what has been laid yesterday. And they've said that this afternoon will not be sufficient.
This afternoon is okay.
So
What
that-
about tomorrow?
That tomorrow.
Let them take the afternoon, then tomorrow they ask questions. But my Lord, honestly, the report which they have had, and my Lord, this is not the first time. You remember the witness we had here, D3, testified on 1-page report. They still said they needed time. So my Lord, this entire thing, the court needs to prevail over the defense. Over the prosecution. Because my Lord, the prosecution can waste all the time. But the court is a court of justice, my Lord. Except if the witness is going back and will be cross-examined on Zoom. That option we will take. But for us to be asked.
What has in the DNA report and what has been testified, some of these matters,
Yes, my Lord.
they are the matters that
Yes, my Lord.
and the prosecution right now has requested to look at what has been brought out in the examination in chief, the report of the witness, so that they can be.
All those matters are in the report, my Lord. All those matters are in that report. The witness limited herself to the report. For you then now to suggest, because my Lord, the next week is not here. This is the 3rd time that the witness will be flying in here, stay here for a week, and then fly back. At that expense, my Lord. Why don't they have the luxury of calling their witnesses at the expense of the state? Accused persons don't. So, my Lord, this court needs to prevail over the prosecution. This time wasting with this will be the 27th adjournment the defense is asking for. 27. Sorry, the prosecution.
Court can prevail on the prosecution within what is for me to sit here and insist that cross-examine tomorrow, and yet they have said they are.
Okay, my Lord, then let's do it on Zoom. Let's pray as a compromise. Let's apply, my Lord, because, my Lord, you gave us a date. She flew in here. Her passport can show. She stayed here for 4 days. We weren't able to hear her. She went back. She flew in here again over the weekend. She stayed here. She's ready to fly back on Sunday. If we don't change how to change the ticket or let her go, and then she will come back here and then get cross-examined for God knows how many days. My Lord, we pray for the indulgence of the court to use Zoom in the circumstances as a compromise, but also to prevail over the defense so that then we can do prosecution, so that we can be able to finalize this matter. No, let me finish. Yes, my Lord. Yes, my Lord. So, my Lord, my plea is that we use Zoom. The witness is available tomorrow. If they are not ready, let's use Zoom, my Lord.
My Lord, if I could just add just 1 or 2 sentences. My Lord, it is not correct or true that there is any ambush in what this witness has told this prosecution. My Lord, the record will reflect that the prosecution themselves supplied here a DNA expert, who are fully instructed as to the science of DNA. We examined and re-examined a prosecution expert. My Lord, the record will also reflect that this prosecution has had the details of this witness and her report for more than 3 months. So the insinuation that there is anything difficult, unknown to the state of Uganda, that this witness has said is a massive embarrassment to the office of the government, actually, because they prosecute in the name of our country. They cannot seek an adjournment on the basis of ignorance.
Absolutely. You cannot.
I think this discussion has degenerated into an area that is not.
Much obliged. Maybe I withdraw that last sentence. But my Lord, the thrust of the point.
Let us preach to whichever we discuss.
Much obliged.